Hello students, welcome to EPG Pathshala. I am Dr. Neelam Gandhi from Zoology Department, Hansraj College. Today we are going to discuss about a module Genome Dynamics in Prokaryotes from paper Molecular Genetics. So students, let us see what we are going to learn in this module. After studying this module, you shall be able to understand that genomes are not fixed entities, but they continue to change, exhibiting new properties. DNA can be transferred to members not only of the same species but also to other species, genera, or even across different domains. Concept and mechanism of horizontal gene transfer in prokaryotes. Then, what is the evolutionary role played by horizontal gene transfer in prokaryotes? Progress is greatly speeded by installing new genes from outside, which get domesticated in the genome of the recipient. Characters conferred by these processes can have long-term implications. Possible applications in the field of public health care, animal husbandry, agriculture, and bioremediation, as well as use of vectors for gene therapy and research. Bacteria evolved 4 billion years ago and continue to exist in all niches very successfully. And there are bacteria which are residing in water and yet they are able to reside in the human bodies. There are bacteria residing in soil and yet they are capable of causing illness in the human body. They are capable of living in such diverse ecosystems. They have become resistant to such a large number of antibiotics developed so far. Earlier held view that genomes are fixed. It is now being revised in the light of many genetic studies. This suggests that there is a dynamic nature of genomes. Transposable elements, plasmids, and viruses, they all play key roles in genome dynamics. And it is the horizontal gene transfer which enables passage of genetic material between genomes by means other than parent to offspring inheritance. This is recognized as a major driving force for evolution of prokaryotes. Genes can be transferred across different species, genera, or even across different domains, which is not possible for vertical gene transfer. New functions and adaptability have been acquired by most gram-negative bacteria as well as gram-positive bacteria due to their resident prophages, which confer a lot of virulence onto these, say for example Staphylococcus aureus, and therefore they pose a great threat, and therefore treating these diseases has become a major challenge. Thus, understanding bacterial evolution at the genetic level is very crucial for devising effective healthcare strategies. Genome changes in bacteria can be brought about by plasmids via the process of conjugation, exogenous DNA via the process of transformation, and bacteriophages via the process of transduction. In the process of conjugation, which was actually discovered by Lederberg and Tatum, and they were using two multiple auxotrophs of E. coli, and the two, when grown together, did not give rise to prototrophs, but when the two were grown together, gave rise to prototrophs. Though the rate of obtaining prototrophs was very, very less, yet it gave a clue that there was a chance of recombination in bacteria. Davis, using his U-tube with a filter, showed that though the two arms of the tube contained the two auxotrophs, because they were prevented from coming in contact with each other, therefore they did not give rise to any prototrophs, establishing the fact that they must come in contact to be able to exchange genes. To further understand the process of recombination, William Hayes conducted an experiment wherein he treated strain A cells with streptomycin and added these cells to the other strain and allowed the two to grow together on minimal culture medium. Prototrophs were recovered. But when the strain B cells were treated with streptomycin mixed with strain A cells and allowed to grow on minimal culture medium, there was no recovery of prototrophs. And this was again a very landmark experiment because it showed that during conjugation, one strain served as donor, the other as a recipient. The process was unidirectional and survival of the recipient cell was necessary for the formation of a recombinant cell. Indicates that the cells which function as donor actually contain an F factor or the fertility factor. They are capable of donating their genes, and this F factor has now been determined to be a double-stranded circular DNA with about 40 to 100 genes and is capable of replicating autonomously, independently of the host chromosome. Genes of the F factor code for the proteins required for the entire process of conjugation, such as the formation of sex pilus, creating a nick, unwinding of the double-stranded helix. It is through the sex pilus that one strand of DNA of plasmid passes to the recipient. Second strand is synthesized in each cell, converting the recipient into the F+. What is actually the basis of recombination? Though it is the Lederberg and Tatum experiment which first of all proved that bacteria can exchange genes, and they did their landmark experiment wherein prototrophs were recovered, though at a very, very low frequency. This was followed by other workers like Cavalier-Sforza, who obtained the HFR strains, and it is the HFR strains which showed recombination at a very high frequency, and that is why named as HFR, high frequency recombination. And it was basically due to integration of the F factor, the fertility factor, into the chromosome of the bacterium. And when this factor was transferred into the recipient, the exchange occurred at a much greater frequency, and this entire process, that is exchange of homologous region of the donor bacterial chromosome with the recipient chromosome, requires the participation of proteins REC-A and REC-BCD, because whenever there is a mutation in these genes producing REC-A and REC-BCD, there is no recombination, and hence it is these proteins which are playing a very important role in production of recombinant bacteria. Shows how high rate of recombination in bacteria can be achieved. This was found when the F factor was found to integrate into the bacterial chromosome, and this led to a very, very high rate of recombination, And this was possible, that is, the integration was possible because of the presence of a common insertion sequence in both the plasmid and the chromosome of the bacterium. Initiation of conjugation and transfer of donor chromosome into the recipient bacterium takes place at the site of origin, and donor chromosome fragment, then having reached the recipient cell, aligns with the homologous region on the host bacterium, replacing the latter by a double crossover event, and the host bacterium acquires new characters. This is all indicated in the diagrams depicted. It shows a cross between F plus and F minus, which is getting converted into F plus. It shows a cross where F plus is getting, it shows the process where F plus is getting converted into HFR, and a cross between HFR and F minus bacterium, and HFR producing a recombinant F minus bacterium. Explains in detail as to how an HFR bacterium is actually formed. This is the recombination event between the IS element or insertion sequence element on the plasmid and homologous insertion sequence element on the bacterial chromosome, involving a single crossover event, and from the point of origin, then the transfer of chromosomal markers can begin when the HFR bacterium starts conjugating with the F minus and starts transferring its chromosomal segment. Shows that though the process of disintegration of the F factor from the chromosome of the bacterium is generally precise, once in a while this disintegration is defective or imprecise. And when it is imprecise, the F factor, while getting disintegrated, also happens to carry along with it a little portion of the bacterial chromosome along with it. And therefore, the bacterial chromosomal markers are carried along with it, and thus the properties of the plasmid, this plasmid, because it is a fertility plasmid, it is now capable of initiating conjugation with another recipient bacterium, is now not only going to transfer the F factor, but also along with it the marker of the bacterial chromosome which it has carried from the donor bacterium. Shows the formation of R plasmid, that is an R plasmid which has gene for resistance against antibiotics, tetracycline, kanamycin, streptomycin and sulfanilamide, ampicillin, mercury contained in a transposon. Use and overuse of antibiotics promotes their evolution, and they can spread very fast to bacterial pathogens, leading to serious clinical problems because our plasmid is also a conjugative plasmid and can be very easily transferred to a recipient bacterium. Integrons are the gene segments present in chromosomes, and they are composed of three essential elements for procuring exogenous genes. One, a gene coding for an integrase enzyme, a primary recombination site at I, and a very strong promoter PC. Integron integrases recombine discrete units of circularized DNA known as gene cassettes downstream of the resident PC promoter at the proximal at I site, permitting expression of their encoded proteins as shown in A, or lead to excision as shown in B. More than 70 different resistance cassettes have been described, and they confer resistance to all beta-lactams, aminoglycosides, chloramphenicol, trimethoprim, streptozotocin, rifampin, erythromycin, antiseptics of the quaternary ammonium compound family, that means a whole lot of antibiotics. Resistance to all of these have been shown to be present on various gene cassettes. Shows the integrons and gene cassettes. Integrons are actually the DNA segments which are composed of three essential elements which can code for an integrase, which have a recombination site and a strong promoter. These integrases recombine discrete units of circularized DNA known as gene cassettes, which are downstream of the resident PC promoter at the proximal site at I site, and this permits expression of the encoded proteins as shown in A or lead to excision as shown in B. More than 70 different resistance cassettes have been described, and they confer resistance to a number of antibiotics and antibacterials, and therefore pose a lot of problem in the treatment of medical diseases. Shows that integrons have become very important because of the features that they have, that they can be present in transposons and conjugative plasmids, and this leads to their mobilization of antibiotic resistance genes. They can travel far and wide and therefore spread to microbes very fast. The other property is that they are capable of capturing antibiotic resistance cassettes, and this allows transcription of antibiotic resistance coding sequences. Shows the landmark experiment which Griffith performed, showing that bacteria can be transformed. The avirulent strain getting changed to the virulent strain by the heat-killed virulent bacteria, wherein a double injection of the rough colony and the heat-killed smooth colony bacteria led to the mice being killed, and the virulent bacteria could be recovered from the dead mice. Therefore, the characteristics of the bacteria can thus be changed. The exact nature as to how the bacterial characteristics were getting changed was then determined by, again, a landmark experiment conducted by Avery, MacLeod and McCarthy, who showed that the transforming principle was in fact the DNA, and this they were able to determine by using a number of enzymes to eliminate the proteins, carbohydrates, RNA, and ultimately if DNA was eliminated, it also eliminated the transforming capacity. Shows that for the process of transformation, it is the exogenous DNA which may be present in the soil, in the water bodies or anywhere. These small pieces of DNA can gain entry into a bacterium which has competent sites or which is in the physiologically competent state and is capable of taking up the exogenous pieces of DNA, and this DNA and this entry is by the process of energy expenditure requiring ATP, and once this piece has entered into the cytoplasm, it is going to align with its homologous region, and by the process of recombination, it gets incorporated into the recipient DNA of the cell, thus altering the property of the recipient bacterium. That the properties of the bacterium can also be changed by another process, and that is transduction, which is mediated by the bacteriophages. This was again conducted by the landmark experiment was conducted by Lederberg and Zinder, wherein they were using two multiple auxotrophic strains of Salmonella, and when they were grown in a common medium in a U-shaped tube separated by a filter, when the filter was of the pore size 100 nanometer, which allowed the passage of particles of the size of the phage particle, there was a recovery of prototrophs, though with a very low frequency, again suggesting that recombination was being achieved. But because this was not requiring direct physical contact, as the bacteria could not pass the filter, this was a process different from the conjugation process, and therefore they had actually discovered a new process of recombination in bacteria, and further experimentation revealed that this was actually being achieved by the mediation of bacteriophages. Shows that as the bacterium gets infected by a bacteriophage, the bacteriophage leads to breakdown of the bacterial chromosome into several pieces, and having utilized the machinery of the bacterium, finally forms its viral particles, and once in a while it leads to the formation of aberrant phage particles, that is, portion of the bacterial chromosome gets encapsulated into the viral head or viral protein coat. So upon lysis of the bacterium, the viral particles which are produced in the lysate, some of the viral particles are actually aberrant because they do not contain the viral DNA, but they contain bacterial DNA. So, because the infective property resides with the protein coat, these aberrant phages are able to infect fresh bacteria, and when they infect other bacteria, they are able to introduce this bacterial DNA and thus produce a change in the bacterial characteristics. On the other hand, there can be another method of transduction, and that is when instead of the phage particle causing immediate lysis of the bacterium, it integrates its own DNA into the phage DNA, and therefore it remains as prophage and it multiplies along with the phage DNA. And it is only once in a while that there is the onset of the lytic cycle, that it comes out of its lysogenic state, and during the excision process, it once in a while carries along with it some of the bacterial genes, and therefore it also results in the formation of specialized transducing particles which contain viral DNA in addition to a little fragment of the bacterial DNA. These are called specialized transducing particles, and when these transducing particles are going to infect another set of bacteria, they are not only going to introduce their own viral genes, but they are also going to introduce the bacterial genes. Transduction seems to have played a very, very important role in transducing characters from one bacterium to another or to the host, and this is due to the fact that many of the bacteriophages are polyvalent in nature, and this allows the same phage to infect many different species of bacteria, thus facilitating gene transfer even in those bacteria which do not show conjugation or transformation. Phages, being resistant to external conditions due to outer protein coat, can persist for long periods of time, and therefore they can participate in HGT. Transduction by bacteriophages can include any bacterial DNA such as linear chromosome fragments, plasmids, or transposons or insertion elements, and enormous numbers of phage particles as well as extremely large numbers of bacteria within the bodies of animals in soil as well as waste treatment plants allow very frequent transduction events. This has led to dissemination of a large number of genes contributing to adaptability of the bacteria to survive under varied environments. Prophages are also responsible for virulence, sporulation, and biofilm formation in a number of pathogenic bacteria, increasing their survival. Thus, we have realized so far that installation of new genes from external source followed by their endogenization has led to a very rapid evolution of microbial pathogens. This is a plasmid which harbors a number of genes including those conferring antibiotic resistance from many different bacterial hosts, and thus treating farm animals with antibiotics leads to appearance of antibiotic resistant bacteria in food items prepared from them. Shows, therefore, applications, formulation of strategies for the use of antibiotics in medical practice, use of antibiotics in farm animals, proper disposal of antibiotics, bioremediation, use of plasmids, transposons, and viral vectors for gene therapy and genetic research. So students, let us summarize what we have learned in this module. Bacteria represent one of the greatest success stories. Having been around for the longest period of time, this adaptability of bacteria has largely been due to the dynamic nature of their genomes. HGT, accomplished by conjugation, transformation, transduction, has allowed bacteria to succeed in all kinds of niches. They can even change their niches from soil to the body, from water to the body, as well as withstand majority of the antibiotics synthesized so far. This has serious implications for the treatment of infectious diseases. Hence, it necessitates a clearer understanding of the genetic mechanisms involved as well as utmost caution for the use or overuse and disposal of antibiotics. Same mechanism may also be harnessed for bioremediation because a number of plasmids confer a property to the bacteria to be able to degrade hydrocarbons, and therefore they can be used for bioremedial purposes of soil and water. Thank you.